Dr. Thompson is going to talk about Erie today.
Dr. Thompson, are you going to talk about your new course, given that statement?
All right. Sure, I can mention that. So actually we had some discussion about having a plant breeding practicum, which is similar to that, but actually organized. It'll go through throughout the year, visiting different breeding programs at certain peak times. But it'll be more of an organized thing where students would take it as credit and you'd write reports on your visits and that sort of thing. But it's also aimed towards, especially like first-year grad students, just to get an introduction to all the different programs. But again, I'll be coordinating with the breeding groups as far as who's available, which times, and that sort of thing. But we're planning to start in the fall, but it'd actually continue throughout that following spring and summer, and students who take it in the fall would get it incomplete until it's finished. So that's the idea. It would be a special topics 6, 8, 9.
So just to clarify, what Brian's talking about is the same thing as your class, or those are two separate things?
No,
No, we haven't.
They're two separate things.
We haven't
They
talked.
haven't talked to each other.
So it's a great idea. We should talk. Okay. So yeah, I'll jump right into my talk today. And actually, what I thought I would do is kind of jump back and give a really broad overview of Erie's plant breeding activity. So I won't be focusing so much on what I was doing. Just near the end, I'll mention that. But I wanted to give everyone a feel for what it's like to be a plant breeder in an international center, especially in the CGIAR system. So I'll introduce that, and this will be really informal, so feel free to interrupt at any time and let me know if you have questions. So I spent about 10 years at Erie, and so I was familiar with the breeding programs. I'm not a field breeder myself, but I was helping with more on the molecular marker side, helping the breeders use markers in their program. But over that time, I got familiar with some of the activities. So just an introduction to the CGIAR system. So this is the Consultative Group on International Agriculture Research, and it's been around since 1971. And it's actually, right now it's organized as a consortium of centers. There's 15 of these CG centers, so we call it CG centers, and each one has one or a few mandate crops. And so many of you may be familiar with CIMMYT, of course with the wheat and maize work, especially with Dr. Borlaug. But there's many more that maybe you haven't heard of. So, for instance, CIAT in Colombia for tropical agriculture, and then CIP for potatoes, and then ICRISAT for semi-arid crops, and others such as World Fish and C4s for forestry. So pretty much anything, and there's also livestock, anything related to agriculture. And essentially this is a way for the international community to really support research that's focused on these crops using really a system of partnerships. And so the funding goes into these international centers, and then the centers work with each country in their respective areas. And so what I'll do is I'll focus more on Erie, but I'll mention that there are 3 centers who have rice as a mandate, and one of them is Africa Rice, which is in Ivory Coast, Cote d'Ivoire, and then CIAT has a small rice breeding group. Although their primary focus is more on, they do cassava and beans and other crops as well. So we do have a relationship or a partnership with both Africa Rice and CIAT. And all these centers generally share, I would say, kind of this atmosphere of interdisciplinary research, because they do have a very small focus on just a few mandate crops, but they have a very wide range of disciplines. And so you'll see the plant breeders working with physiologists, working with even socioeconomics, and then GIS, so geographical information, agronomy, across the whole board of all the different disciplines focused on a single crop. And so at Erie, this is based actually in the Philippines, about an hour from Manila, and it was founded in 1960, so it was actually one of the first of these international research centers, and actually founded by the Ford and Rockefeller Foundations. And at the beginning, they had, it's just right at the beginning of what led to the Green Revolution, essentially CIMMYT and Erie kind of leading the way for wheat, maize, and rice. And essentially Erie's mission, as you might imagine, is to reduce poverty and hunger, improve the health of rice farmers and consumers, and ensure environmental sustainability of rice farming. And the way they do that is actually through collaborative research and partnerships, largely with the national centers. So generally each country will have its own national agriculture research and extension system. So for instance, Erie works with at least 20 countries, and they all have rice breeding programs. But there's certain things that can be done when you have a consolidated group that can essentially go deeper than each national system could go. So there's some synergies that the idea is that you bring together strengths from many countries and you can make faster progress. So we do have, or at Erie, they have about 1,000 research staff, including about 100 scientists and 120 graduate students. Generally, roughly half of them are actually from the University of Philippines, Los Baños campus, which is their agriculture school in the Philippines. And the other half generally would have their degree awarded by their home institute, and they would do research at Erie. So there's quite a few visiting graduate students on campus, even though Erie itself doesn't award any degrees. So just many people are wondering, how are these centers funded? Just to give an example of Erie, this is in 2014, they had a budget roughly $100 million annual research and administrative budget. And the source, largely from the CGIAR fund. This is actually, they have a fund council where the donors actually kind of throw money into a pool, and then it gets directed into research programs. Largely from World Bank and then many of the national developed countries. And then some of these, of course, many of the countries do more direct funding, like bilateral agreements directly to Erie without going through the fund. So generally when it says national governments, we're talking about like European Union, Japan, US, Germany, and many of these developed countries giving international funding for research and development. But there is quite a large chunk now from philanthropic foundations, largely Bill and Melinda Gates Foundation most recently, who's really strongly supported agriculture research in the past, I would say, 5 to 10 years. And a little bit from private sector. So if you look at the structure, so Erie's structured mainly around 4 divisions. One is plant breeding. That's been traditionally their largest division. It used to be actually combined with the genetics and biotechnology. They recently kind of divided those two. And plant breeding contains all of the breeding programs, and essentially there's 2 other units as well, Seed Health Unit and then Grain Quality Nutrition Center within the plant breeding division. And then genetics and biotechnology is more the upstream research. A lot of the gene discovery work goes on there, and it also contains the Genetic Resources Center. So that's the Erie Gene Bank that I'll talk about. It contains a lot of accessions of rice. And then there's a smaller center called the C4 Rice Center, so trying to convert rice from C3 to C4 photosynthesis. And then they have quite a large division for crop and environmental sciences. This includes agronomy and crop management and also climate change, and those fields. And then they do have social sciences as well. So a lot of this is based more towards kind of impact assessment, but also policy. How does, rice is a very important crop around the world, so how does that affect, rice policy, and how does that feed back into research priorities? And there's, as well as a training center. So Erie does have an educational mandate, but it's quite smaller than what you'd find at a normal university. But you do have, we had actually 15 country offices, including 2 research hubs, I would say. Yep.
So on your budget there, if you say half the students are from Los Baños, Philippines,
yeah.
Is that included in that, or are there a lot of sort of in-kind support that come from the community?
Right. So that would be, because this would only be budget that's actually funneled through Erie. But generally, if they're awarded a scholarship, if it's awarded through the training center, that would be included as well.
So Erie's often supporting the grad students who are going to.
That's right.
Okay.
They do have some support, not a lot, but there's some support available for that. So it depends where they're studying, and they do. In the past, they've had quite a few kind of scholarship programs. Recently, I should mention that recently this budget has actually dropped significantly in the past 2 years. And so they're going through kind of a budget crunch at the moment across the whole CG system. And so these things, it's kind of cyclical, and this has happened before. I mean, with the food crisis back in 2008, everyone's worried about, price of rice and these major commodities skyrocketing. And then the funding is really nice. So Erie went through a growth phase. And then now, everyone forgets about that, so the funding drops. So it's going through, so it's actually much less at the moment. And they're also, the CGIAR system is going through a change process where they're trying to reform the structure of how it works. And so they're right in a transition period. So once they get past that, hopefully the funding will improve.
So before you leave this slide, can you spend 30 seconds, when they cut the budget, did they cut the C4 Rice Center?
Yes, so that was one of the ones that got hit pretty hard. But that actually is interesting because it's actually mostly from like Bill and Melinda Gates Foundation, and it was a restricted project. It didn't go through the CG system, but it was cut for other reasons, essentially.
It's not, people have been looking at that since I was in graduate school.
Yeah,
Surprised
it's
to
a
see
tough.
that rearing its head all the time.
Right. It's definitely a long-term project,
Big
and we'll
time.
see. They've made some progress, but it would be another 15 years before anything really happens, I'd say. So, any more questions? So just to let you know, like, where is rice grown? And Erie's mandate is largely in Asia because that's where the majority of rice is grown. So if you'll see here, actually, at the very local level, each of these green dots is actually where a significant portion of rice is grown. So in certain countries, like in the Philippines, we have kind of the rice basket here in Luzon. In Indonesia, Java, if you travel across Java, you'll see that rice is just grown pretty much everywhere in Java. But again, you'll notice that if you take on a larger scale, it's really China is just massive rice production in India as well. And you also notice here in Bangladesh, and then this is Myanmar, Vietnam, Thailand, these are all major rice producing countries. And Vietnam and Thailand especially are also major exporters as well. Whereas most of the other consumption is actually for self-sufficiency in many of these countries. And so, and of course, also the traditional, like, more temperate rice, you'll see Japan, South and North Korea, and then northern China, which is more the sticky rice, where you have the sushi and that sort of rice. Whereas down in Southeast Asia, it's kind of intermediate, like your Thai jasmine types. And in India, there's a very diverse different types of rice in India. But you'll see it's a massive area, and there's many differences between the countries. So as we're talking, you'll see that it depends where you're focused, which region, but also what type of rice, whether it's irrigated or upland and these types of things. So Erie is actually working across all of these areas. And just to give you an idea of kind of historically where rice yield's been, and this is from essentially the Green Revolution, so mid-60s with the release of IR8. Essentially, these are kind of the progress that's been made. Some of the reasons for it, on the bottom, these tend to be, it's more the genetic and plant breeding advances, and then on the top is more of the agronomy and crop management. And essentially, we still follow the rule where it's mostly 50-50 between the genetic and the management, providing this pretty steady gain in yield improvements due to many reasons. Early on, the Green Revolution, mainly due to the semi-dwarf. After that, a lot was focused really on the disease and insect resistance, and then moving into focusing, getting to grain quality, starting a hybrid rice program, taking introgressions from wild rice, and now more on gene discovery and kind of precision breeding and using markers. And currently looking more on yield potential per se, is trying to break the yield plateau that's essentially since this time, you've seen this almost level off even less. And what it equates to is it's about 1% genetic gain, whereas if you look at the demand, we really need more like 2% per year. And so this is something that, and while I was there, especially, a lot of discussions on how we can increase this rate of genetic gain, because if you continue at this rate, it's actually not going to be enough by 2050, when we have 9 billion people. And in many areas, rice consumption is still increasing as well. And so, since that's been a concern, but at the same time, it's also, if we didn't have these yield increases, we would have used a lot more land as well. So now to kind of look specifically at Erie's breeding program, what is the structure? And it's really structured in, they have 7 different, what they call breeding pipelines. And you'll see these are broken down. There's one that's focused just on hybrid rice, and this is irrigated largely, and across both South and Southeast Asia, which are the 2 primary target areas for Erie. And after that, you have all of these are inbred programs. So Erie is still largely inbred rice breeding for many reasons. And the biggest is, in many countries, hybrid rice is still a relatively small proportion of rice grown, with the exception of China. And so the inbred irrigated Southeast Asia program has been traditionally the largest program. This is the one that's produced, say, like IR8 and IR64, these really mega varieties that we call them, grown across massive areas of land in Southeast Asia. But then South Asia as well, so more on the irrigated program has made some advances recently. But then they also divide it by rain-fed as well. So you'll go, if you're in Asia, you'll notice that many areas, they do not have irrigation. It's just the infrastructure is not available. And so there's very large areas that are just rain-fed. And so there's generally a wet monsoon season, and so they'll be growing rice and just depend on the rain. And so there's a number of different traits that are needed, and there's different breeding pipelines of both rain-fed Southeast Asia, South Asia. And then in Africa, they have they've essentially combined those two. They think, and actually Africa is an interesting case. Rice is still grown relatively minor crop there, but it's growing very quickly, and there's actually quite a bit of land available. So it's more, there's kind of unique challenges in Africa, but that's something that recently has been a really big focus as well. And then lastly, a small program on actually japonica rice for the tropics, which is an interesting one where they are growing kind of the traditional temperate rice, but in the Philippines. Okay, so Erie, they have the breeding pipelines. These are the field breeders with large programs. But really, a large proportion of the scientists are focused on what we call trait development pipelines. And so these are all the traits that the breeders need, and essentially the upstream research that's needed to identify new donors, genetic donors for these traits, to identify what are the QTLs and genes, and really to develop kind of the pre-breeding lines that the breeders can very quickly introgress in their programs. And there's, just to name some of them, so yield potential, again, looking at how to increase yield per se. So this is, sink-source relationships, photosynthesis, looking at many different aspects of that. There's one on anaerobic germination. So this is what Septi was working on as far as having early seed establishment in areas that are flood prone, where they don't have good drainage. Moving on, so biofortification for healthy rice. Green Super Rice was a program essentially trying to develop rice that uses fewer inputs but still has high yields. And disease resistance, of course, has always been important. Moving down to grain quality, flood, and heat tolerance. And then nitrogen and phosphorus use efficiency has been important. And you'll notice that actually the irrigated programs, these are the ones that tend to be most important. And then for the rain-fed, it's more in the stress tolerance. So salinity and drought are really huge constraints across many areas of rain-fed rice production, especially in India and Bangladesh. Yeah.
On the hybrid rice, what is the yield potential from hybridization, and what's the system?
Yeah, so in rice, they've generally seen about 15 to 20% really kind of extra yield potential, depending on what you look, which programs you're looking at. But it's a cytoplasmic male sterility system. Almost everyone's still using a 3-line hybrid system. There is some work on 2-line kind of thermosensitive sterility as well, but that's still in the early stages. And essentially many people are using very similar male sterile lines, actually. So that's one issue, is that a lot of the germplasm has a common kind of source. So that's another thing that people are trying to expand that.
Dangerous.
Yeah. So, and that's, in fact, many people are doing hybrids within indica, because there's several subgroups of rice. There's a few programs doing indica by japonica, but it has other challenges as well. And so it gets complicated, but I would say it's not a huge kind of increase like maize has been. So rice, again, they don't really have the heterotic groups as well defined, and so there's some research to define those better, and as far as the compatibility goes. I would say we're quite behind still in that. But here in the U.S., I mean, hybrid rice has grown very quickly. In some areas, really, it's about 50% hybrid rice production now. A lot of the lines from Rice Tech have been quite successful. And so just on the side, and here, a lot of the public programs now are investing more in hybrid rice. Arkansas has hired a breeder, hybrid rice breeder, and then as well in Beaumont as well. That's a good question. Any others? Yeah.
Which of these areas apply the sub-1 gene more? Or is it introgressed in a lot of the varieties in that area, or is it more?
That's a good question, because generally sub-1 has been rain-fed for South Asia. Really large areas of India, Bangladesh, it's really needed that.
Because of the monsoons, right?
Yeah, that's right. And many areas, they just don't have drainage, and so you'll actually have complete flooding for up to 2 weeks, so this flash flooding. And I'll show a slide on that as well. And then just the end, these are not as important generally because the cold tolerance in most tropical rice is not an issue, but there's certain areas in Africa or high elevation, and even in northern Bangladesh, where you do have cold, because they plant it early in the season, and so they do need some cold tolerance. But I would say heat tolerance, which is here, is there's a lot more emphasis on heat as well. Okay, so that's essentially the main structure, but at Erie they also put a lot of emphasis on cross-cutting teams, and so this, instead of having everyone do their individual own independent programs, they'd like to have some support across breeding programs. And on the kind of logistics side, there's quite a bit of support as far as hybridization. There's a central hybridization team where everyone nominates their crosses, and then the team will do the crossing. As far as field and greenhouses, disease and pest screening for each disease, the breeders will just submit the lines, and then the plant pathologist will screen them. Same with quality, and then breeders seed production. Generally, this is focused to get it more efficient by using a central resource. And in many ways, that's also applied towards the upstream research as well. So there's now a more organized trait discovery research team developing the kind of specialized genetic stocks, and then moving into the gene discovery part. Traditionally, this has actually been done, each group kind of did their own, and so now they're trying to also centralize that to make it more efficient, and then with the molecular marker applications as well. So I'll kind of run through some of these just to give more of an overview. And so the idea is that across the breeding programs, then the breeders don't have to worry about these kind of logistical issues. In some ways, moving more towards a kind of a private sector approach. And so just going back to the history, so most people have heard of IR8, the first variety of the Green Revolution, and as well as IR64, that's kind of the second one that we really highlight because it was grown in such a massive area of land. So at its peak, it was grown on over 47 million hectares in Indonesia, this single variety. So it's really amazing how a single variety could be so popular. And there is some impact assessment, not just these varieties, but across all of Erie's varieties. Really economic benefits over $1 billion a year from this study, looking across mainly like Indonesia, Philippines, Vietnam, these countries where the improved varieties really have a huge effect. And going back to the history, so it's interesting that, so IR8 is actually the 8th cross made by Peter Jennings. Early on, he was Erie's first rice breeder, and followed soon afterwards by Hank Beechel, Dr. Hank Beechel, who spent many years actually in Beaumont, really kind of leading the way for the U.S. Rice breeding. He moved to Erie at that time, and so they developed essentially this IR8 that started kind of the Green Revolution in rice, mainly because it had the semi-dwarf stature from D.G. Owujin from Taiwan. So this was an interesting case where it's actually a single gene that allowed this massive yield increase, mainly because you can add fertilizer and it doesn't lodge. So it withstood that. So you can essentially have higher production. But one thing to note is that there's only 3 land races in this pedigree. And we'll kind of compare that to some of the more modern varieties. I'll just also mention that this was the 8th cross. In 2012, Erie's hybridization program went by their 100,000th cross. And so at Erie, they do roughly 2,000 crosses a year. And so they continued this, and across all the breeding programs, they're doing quite a few crosses. But just to note that here's a pedigree of a more modern variety, IR72, and it actually has 22 kind of ultimate land races in its pedigree. Many of these to bring in disease resistance, insect resistance, and different grain quality traits. And so Erie traditionally has been completely pedigree-based breeding, and essentially just going in and trying to bring in what's needed to create improved rice varieties. And so this is something that they essentially tracked the pedigrees this whole time. And so this is kind of the traditional method that has been used for many years. The pedigree method, going through, and actually in each of these generations, a lot of the selection is done by eye, by the breeders out in the field. And it is pretty amazing that they've been very successful because of the art of plant breeding. Those of you who are field breeders know that can be very successful. And so this is observational yield trials, and then replicated, and then into this multi-environment testing. Now, one thing though is it is relatively a long breeding cycle for rice, and rice is a crop that you can get up to 3 cycles a year if you're doing kind of a rapid generation, whereas this breeding cycle has been relatively slow. So recently, around 2012, they restructured the breeding programs to make it faster. So they wanted something that you get faster results and increase that genetic gain per year. And the other thing is that the footprint is quite large. So just for the inbred irrigated Southeast Asian program, they're using about 15 to 20 hectares per season at Erie, just for the yield observations, not including the multi-environment testing. And so it's a very large program. And this is essentially kind of the new scheme that they shifted to back around 2013, essentially trying to again, speed up the breeding cycle. And they do that through rapid generation advance, essentially single seed descent. In rice, there aren't really very good double haploid programs. No one's really done it on a production level. There is quite a bit of work on the research level, but it's not really efficient enough. And so it turns out that the single seed descent is pretty efficient. So you can do, they've essentially created a system where they can do 100,000 lines per year. And going through very rapidly fixing them, and then you do essentially observational yield trial unreplicated, and then partial replication of preliminary yield trials, and starting multi-environment testing much earlier in the season than when it was done previously. Previously, they waited to the very end before they sent them out to multi-environments. So trying to get the multi-location earlier, the breeding cycle faster, and then starting to use markers and hopefully genomic selection at some point. So in this case, shortening the cycle to 4 to 5 years so that these can go back into the crossing program much faster. And that also reduces the footprint because the RGA is really a more dense production. And so here's essentially the single seed descent in the greenhouse, very large scale, so trying to get this to where it's a production facility for rapid generation advance, essentially replacing what most companies would do with a DH production facility. And of course, there's a lot of logistics involved as far as tracking these. And Bert Collard was the one who was leading this effort at Erie. But I think they've come up with a pretty good solution for that. And then really important is the multi-environment trial. So this is something that, in the past, they didn't really focus on as much. Many times the breeders had their own kind of area that they were looking at, but they wanted to expand this like most companies do. And so essentially expanding in the Philippines, but also across Southeast Asia. So they go through several different multi-environment trial. For instance, they call it MET0 and then MET1, MET2, essentially where they go through multi-environments and then they take a subset of lines into the next generation, into essentially larger, more highly replicated experiments at each one, but fewer lines by MET2. And so this has recently been expanded as well. But I'll mention, so these are really high quality research plots that Erie has been leading, but there's also quite a bit of germplasm exchange on a broader level with the national programs. So this is something they call the International Network for Genetic Evaluation of Rice, or INGER, and essentially this is where they would take release varieties and the best breeding lines and essentially just freely distribute it to anyone who's requested it, who's willing to grow out these lines. So there's essentially packets of, say, 50 lines for a particular environment and ecosystem, and this is just sent around the world. And so this has been quite successful, and it's not just Erie lines, there's nominations from other countries as well, to get a much larger scale multi-environment data. But at the same time, this is on a volunteer basis, so you don't really get the high quality data that you might expect if you were doing it, organizing it yourself. But it is quite useful, and they do have, Erie really has a massive amount of multi-environment data, for instance, for G by E type. But a lot of this, I think they could probably do more with it as far as extracting useful information from that.
Michael, that's line testing per se, or is it, are any of these in any form of hybrid trials?
Right. So the hybrid program has an independent system, but in this case, what it is essentially is advanced lines that national programs can use. Either they can release them directly if it's suitable for them, or they use it in their own breeding programs. So in the past, actually Erie's been very open with their germplasm and breeding lines, where they essentially just give it away to anyone who needs it.
So they're evaluating it based on how they would want it.
That's right. So everyone will take it, they'll evaluate it on what they need, and they'll take what is useful to them. And we only ask that they would, kind of help Erie give back some data, or at least let Erie know that, okay, we used this line in our cross, or we released it because generally each national program will rename the varieties, and so no one knows that they're Erie varieties actually. So they're, in a way, Erie wants that kind of brand recognition as well. So there's been some issues with that. But as international public institute, I mean, that's their mandate actually, is to help the national programs. So I think this has been one mechanism where they do that. But more recently, there has been discussion as far as the IP protection, and Erie has also hired IP people to think about, okay, certain lines that they consider high value are more sensitive, that they may want to license, freely license to some companies, but in some areas actually license it to the private sector. So there's some issues with that as well.
Michael?
Yeah.
So what are the issues with the multi-environment trial, the phytosanitary, and getting seed to all those countries?
Yeah.
I'm guessing most of the students don't know what a pain in the butt that can be in.
Yeah, and rice it's huge. It's extremely difficult. So this was Ed Radoni was the head of this program, and that was his kind of skill, was getting rice. They had it planned down exactly to when you can send it, when it had to be applying for these certificates, and essentially it is challenging, but they made it work. And so the only problem is, well, I don't know if it's a problem in some cases, but in India and China, essentially you can send it there, but you can't get anything back. So we never actually set up any actual breeding in those countries because we couldn't get, even if it's our own line that we make crosses, we couldn't get it back.
Is that a disease thing or is that an IP thing?
That was an IP thing more.
So is there disease issues that certain countries have banned stuff from the Philippines, for instance, or?
Generally, since we have very good kind of seed health group, they would essentially validate everything that it was clean going out, and everything coming in as well would go through quarantine and be verified. And so there hasn't been issues. I haven't heard of any major issues where actually a disease would stop that process. They had the process in place to make sure there weren't any problems, and it essentially was working very well. So it didn't impede the sharing of germplasm, except that it would slow it down. I mean, you'd take 3 months or something to go through this process. But that's a good thing to know. I mean, you can't just ask someone to send you rice seed, especially here to the U.S., without going through this very detailed process. So. In some cases, DNA as well. And we found in the Philippines, their phytosanitary, it was still applying to DNA as well, so we had to. It was very strange. But I think they've improved that recently. But it wasn't easy. There's some challenges to working in an international institute, and that was one of them. So moving on to the range for the irrigated program, some of the recent kind of thrusts have been really in flood tolerance. So this is the sub-1 story that I'll talk about. But also looking at really early flowering, very short duration. In many places, for instance, Vietnam, they actually get 3 seasons in the field, 3 rice crops per year. Instead of most places in Asia, you actually have 2. You have the wet and the dry season. But if you have very early flowering, you can actually get 3. And sustainably, you can grow 3 crops in the same place over and over again without having a fallow field. Because of the irrigated system, there's quite a bit of research in the long-term cropping of rice that you can actually have it sustainable as well. But so at Erie, actually, most rice is hand transplanted. It's actually manually transplanted, even though the research farm, is like 400 hectares or something. But they have really hundreds of people out there going and manually transplanting rice. So there has been a shift now to try to get to direct seeding, which is done, in the U.S. and at most other, like in South America, and even now in India, increasingly has been direct seeded. So this is, planting drill seeding, either dry direct or wet, kind of broadcast seeding rice. And then, of course, trying to get water saving. So this is a push for alternate wetting and drying and various drought tolerance research as well. So these are kind of the more recent areas of research. And I mentioned it briefly before, grain quality is very important in rice. And this particular area, they've done a lot of studies across countries looking at consumer preferences, because every country, essentially, they have a different preference. I mean, rice is a very personal thing in Asia where, this rice it has a very unique preference to each country. And so if you look at, these are like basmati, super long grain versus, just long grain, and sometimes, very short or bold type rice. It depends where you are. And so the breeding programs generally will have this market segmentation that they do, research into consumer preferences, but also milling preferences for the target countries. And a lot of this is done, at the national level, but even subnational, for instance, within India. And so the head of the grain quality group, Dr. Nesse, has done a lot of work in grain quality and consumer preferences, and also in the eating quality. So rice, the grain quality, a large part of it is how it tastes, the texture after you cook it. And a lot of those, they haven't really discovered the genetic basis yet. So someone asked about hybrid rice, and so this is actually Erie's really initiated a hybrid rice development consortium, which is a public-private partnership, and has been quite successful now to have roughly 75 members, about half public and half private. And so you'll see across these are the different countries for the members of this consortium, a huge interest in India and of course China, where hybrid rice began, but also many other areas of Southeast Asia that are really interested, in growing hybrid rice. And so this is something where Erie has a central hybrid rice breeding program, but the countries, in this case, if they're members of the consortium, they can actually go to Erie and select lines that they think would be promising and then use them in their own hybrid rice programs. And so this is something that it's germplasm sharing, but it's also they have a testing program where they'll nominate, each country will nominate their lines, and again, they'll be tested across multi-environments. And so this is independent of the other one I showed, where this is only hybrid line testing. And it's been successful, I think, because it is public-private, because a lot of the investment recently in hybrid rice has been from the private sector, especially in India. It's a huge, but as well as Southeast Asia. A lot of the big players from Bayer and Pioneer and others have really invested quite a bit in hybrid rice in the private sector. So that's really Erie, I think, is trying to work and trying to find the way as where does a public institute fit in that scheme. So coming back to right here, this kind of basic research. Now I'll shift really to the upstream work. And sorry, this is, like I mentioned, rain-fed rice has a lot of challenges. And really you can imagine if you don't have irrigation, drought is a big one, but many along the coastal regions will have salinity in the dry season. You'll actually have the saline water travel upstream of the river. So the irrigation that you're using for your field will become salt water. And so that rice is extremely sensitive to salinity. And so that's a big problem. And then the flash flooding. So submergence, of course, rice grows really well in water, standing water, but when it's completely submerged, it can't get oxygen, and after about a week, it'll die. So in some cases, even less than a week. So if you have a flash flood that completely submerges the field, you'll lose your entire crop. And so that's where the sub-1 gene has really kind of made a huge impact for flooding. And then I mentioned heat tolerance as well. Heat, generally, it's the reproductive stage. Pollen sterility, that's been the issue with that. Okay, so early on, Erie invested in looking for novel genetic donors and then mapping the QTLs. And for certain traits, they did find major QTLs. Sub-1 is the best known, but as well as salinity, saltol, and essentially for seedling stage salinity, you can see it's just a huge effect for some of these major QTLs, and also even phosphorus uptake, some of the nutrition as well. And so what they decided at that time, a few years back, is actually to use marker-assisted backcrossing to transfer the major genes. This is because rice is in some ways unique in that it has so many major large effect genes that this has been quite successful. So it's just a traditional backcrossing. But in this case, you're selecting for very closely flanking markers to decrease the integration size so that you don't have negative linkage drag at the donor site, but also cleaning up the background more quickly, so you can actually stop at BC3 with a clean background if you use enough, a large enough population and enough markers. So very rapid marker-assisted backcrossing. And this has been very successful for, again, the sub-1 story. So what we're looking at here, actually, this is work done by Dr. Septi, is essentially the lines that survive. This is a field that was under flash flooding for over 3 weeks, which is complete submergence. Anything that didn't have the sub-1 gene, essentially it completely died. There's just a few kind of stragglers there. And these plots that have sub-1 have really, really strong survival. And so essentially giving what is either almost no yield to a pretty standard amount of yield just because of a single major gene. So that's why sub-1 was such a large impact. And in cases where the flooding wasn't as severe, it still would give you, double the yields in many cases. Again, flash flooding. if you have, there's other types of floods, such as stagnant flooding. I think you've heard Dr. Septi give a seminar a while back, but then it's only good to up to about 2 to 3 weeks. After that, these will also die eventually. But recently, the Bill and Melinda Gates Foundation has really invested quite a bit to scale up the sub-1 varieties across South Asia. And essentially, they've reached over 10 million farmers at this point with the single sub-1 gene that was backcrossed at Erie and disseminated across the national programs. And so, like Bill Gates came to Erie kind of to see the progress, and it's pretty exciting to see kind of the interest in sub-1 and how that's kind of amplified. That effect has really helped, investments in Erie and helped make their essentially more successful to reach the farmers with what's needed. And it was a good example as well of molecular markers making an impact. Before this, really everything had been traditional breeding. And so they've tried to essentially duplicate that with drought, and of course drought is a different story. There's not a single major drought gene, there's like 50 or 100 or something. But they were successful, this is Arvind Kumar's work, and even Gary Allen early on, finding really a large number of drought genes, and some of them were quite a large effect, which was surprising because they thought they'd all be very minor effect. And so this is also in progress. And I'll just kind of move on a little bit to more recent work. They have invested, again, like everyone else in phenomics, kind of high throughput phenotyping. In this case, more of they call it the field scanner, where they just run a tractor across these large buns and then have very wide boom. Of course, in rice, you can't run the tractor through the field. So having the sensors and essentially having it, like this, so that you'd have data across the field, and then it would just go up and down rice fields. This is something that the breeders aren't really using at this point. It was more on the drought physiology team so far, looking at drought stress, can it be temperature, that sort of thing. But I think it will be expanding that theory as well. They aren't doing much on UAVs yet, but this has kind of been the focus so far. So now just kind of wrapping up, looking at the trait validation. This is some of the work I was more involved with as far as how to use QTLs in a breeding program. And really it starts with the gene bank. So I mentioned the Erie Rice Gene Bank collection. There's actually over 124,000 accessions. Of course, there are some duplicates in there. They save everything. They never throw away anything. But these are individual accessions. So you can actually request them. You can study them. And essentially it's a massive wealth of genetic diversity that we don't know how to use for breeding, because of course the breeders can't test them all. So really some of the different strategies, one way is looking into this magic design, this multi-parent advanced generation intercross, where I was involved with this group also, using essentially 8 parents. We had an indica, 8-parent magic, japonica, 8-parent, and then we crossed them 16-parent intercross, we call the global magic population. And in this case, we knew that these parents had useful traits, but we just, using this both to map the genes, but also to get those novel combinations that may be useful for the breeders as far as having that mix of all the beneficial genes and very quickly being able to find out which ones are useful. My own work was more in the markers, so how do we use markers to introgress these genes? I mentioned marker-assisted backcrossing. In the pedigree, just using pure straight marker-assisted selection for major genes, and then moving near the end, we started some tests with genome-wide selection. Essentially, I headed up what was called the Genotyping Services Laboratory, and we provided the service to the breeders, but also to the research groups at Erie. And for instance, in 2014, we processed over 30 million SNP data points and about 10,000 DNA samples at that time, but we had quite a good team. Essentially using SNP data, SNP genotyping for high throughput genotyping, and going through this process. So we did invest more in tissue sampling, using this leaf puncher that would automatically record the individual identity of each leaf sample, and go into automated DNA extraction with a 96 format, which could then go into one of the genotyping systems. So either Fluidigm or in this case Infinium, using a chip developed at Cornell, a 6K chip. And this was kind of the workhorse that we were using most commonly. It had about 4,600 high quality markers, and using this really initially for genetic diversity. So you can see the 5 different subgroups of rice, and even breaking down within indica many of the different subgroups within indica. And so we found that was really quite useful for the discovery, but for breeders, it's most useful really when they're trying to compare new germplasm with what they're using now to see how does it relate, but also trying to move towards genomic selection. The only problem is it is still a bit too expensive to use really mainstream in the breeding program. So we did look into GBS as well, so I was involved in this study for bacterial blight resistance, but actually using GBS service at Cornell, which works quite well, so we get about 40,000 SNPs in rice with a highly multiplexed GBS, which is really roughly the same price as the 6K chip, but a lot messier data, so there's a lot more work that needs to be done on the data side. And then for the low cost for the breeders, the fluid ion. In this case, we're using Caspar, and roughly similar systems where you have trait-specific markers, so something that a single SNP or several SNPs could tell you, okay, this one is resistant for bacterial blight, and this one is susceptible. And that's really, by the time I left Erie, we had worked to validate about 20 of these gene-based SNP markers for the breeding programs, and that work is continuing. There's a fellow, Dr. Tobias Kretschmar, who's continuing this work at Erie, and now I'll be doing some of this work here in the U.S. in the U.S. rice community. And just kind of to leave you with a few future directions. These were future directions both at Erie, but also I think relevant to our research. One is really developing more on the breeding informatics side. This work being done by the Integrated Breeding Platform, but also this new initiative, it's the Genomic and Open Source Breeding Informatics Initiative being led out of Cornell, where trying to really get to high-density genomic analysis kind of integrated into the breeding programs. And it's really quite a big challenge. I think there's some ways where we can essentially learn from what they're doing. A lot of it's based on imputation and tracking major details, but also doing prediction-based modeling. And then really the gene banks, this personal interest of mine is how do we better use the gene banks? And so one initiative at Erie, they're trying to essentially sequence the entire gene bank. They've started with 3,000 rice genomes, and now they'll be moving on once they get more funding to others. And then the DISH-seq initiative, essentially, this is a kind of a global effort to essentially make the process more efficient. How do you explore the gene banks? How do you get the informatics available in a way that breeders can actually use gene bank information and make it accessible to everyone? So that's something that we'll be keeping an eye on as well. And so, oops, sorry. But just to wrap up, I just had one more slide. Go back. So this is really the work that I'm working on now, is trying to develop kind of a system where you have all these major genes that are cloned in rice. Many of these people have published some SNPs, but they've never been validated on different germplasm in different environments, different backgrounds. So trying to consolidate all that information as far as the size of the effects, the allele frequency, validation data on these SNP markers, and then develop a tool, I'm currently calling the Rice SNP Advisor, kind of an advisory tool for breeders. So they can go in, they essentially input their germplasm needs, their trait profiles, what kind of alleles are they interested in, and then essentially the SNP markers then would be available so that they can use that in their own breeding programs, and using such like a Caspar system to get that running, at least for the large effect major genes. And so this could be done for introgression, but also for profiling, which is a bit of a bigger challenge, but if you have some unknown germplasm, you want to know, okay, what genes are in there already, using these kind of SNP haplotypes or gene-based SNP markers to do that. And so that's something that I'll be kind of working with here as well. And so lastly, I'd just like to leave it with genome editing, because of course that's really the future, we think, in rice as well, as far as how can we use this for breeding. Of course it's a great discovery tool, but for breeding, what we're looking at is which genes, if you knock out the function, you get the desired phenotype. So SD1 was a knockout, also the fragrance gene. But more importantly, if you replace alleles. So in rice, we have a lot of exotic germplasm that breeders can't really use very efficiently because the negative linkage drag, the negative traits. So if we know there's a useful allele, say in a wild species, instead of backcrossing it, why don't we just take that allele and replace it with CRISPR-Cas9? And we could do that, say, not just one gene, but say 10 genes in a multiplex genome editing process that essentially gets rid of all of that backcrossing and selection. So that's really what we're kind of, the future that we envision as far as if we know the causal variant at these major genes, or even if we just know the allele that we want, being able to use that more quickly in a breeding program. So with that, thank you, and I think a little bit of time for questions. So, yeah.
Yeah, how about the is with transformation work? You already mentioned that CRISPR Cas9.
Yeah.
And where is the golden rice stands?
Yes, that's a good question. So Erie does have a very active plant transformation facility. Dr. Inez Slomaladin is the head of that. And Inez, she's from Indonesia.
Oh, it's you.
Yeah, and then so essentially the golden rice, they had a few kind of setbacks where they changed events kind of midway. And so because some of the events didn't have high enough effect early on, and then later on they changed because it wasn't stable. And so that program, again, had some delays. But essentially what they're saying now is they're back on track as far as looking for release in Philippines, Indonesia, and Bangladesh. But they won't give a deadline.
Is it one or two? Syngenta has the two. Golden rice.
Yes, golden rice.
One, Puerto Rico's one, and Syngenta produced golden rice two.
Yeah, from Syngenta.
Is it?
It's the one that had a higher
Yes.
higher effect. That's the one they're going to use, and it's from maize actually. So, and then yeah, so that's now at the regulatory phase. You know, testing and trying to get through that deregulation process. So it's kind of their first attempt to push a transgenic rice, and it's a very sensitive issue for rice because many countries, of course, because they eat it whole as a whole grain, and it's such an important aspect of food that it's been quite controversial. But it's interesting that and it's such a useful trait to have the provitamin A. So we're still following that story, but it will be a few more years yet.
Well, I was in Bangladesh a few months ago. I had a talk about my wife.
Yeah.
And I talk a little bit about GMO. It seems to me is most of the people against it.
Yeah, that's certainly true in many areas of Asia.
And as you said, it is moving towards Cas9 system, and it would be GMO then.
Yes, well, the hope is that
But it's
actually
from
it won't
rice
be
to
GMO.
rice, yeah.
Well, I mean, like USDA has considered it not really GMO essentially because it doesn't leave behind any fragment. You only have the change, the mutation. But that hasn't been, hasn't gone through all of the different various agencies to declare it officially, non-GM. And of course, the public may still say, we don't want it. So, what else? Last questions, yeah.
So one of the things you mentioned is that rice has these major genes, and we don't see those in maize. You sort of see them in sorghum. Why do you think that is?
Yeah, that's a good question. 'Cause in some ways, it could be. I mean, it comes down to like the genetic architecture, and maybe even the history of rice in some ways. Because we see, and even though we have, a single major gene like SD1, but actually there are multiple variants. There's different polymorphisms that lead to the same phenotype. And so possibly it could be, I mean, rice is a very simple diploid. There's very little kind of ancestral duplication in it. I mean, it's pretty simple. There's generally, a lot of gene families, they're not huge gene families. There's fewer. I mean, that could be part of it. But I really have no good answer. It'd be good if someone can, look into it.
I think
more.
rice is one of the few species crop species that has been shown to have multiple domestication events.
Yes, that's also true.
Does that play
well,
in?
that's still controversial, but most people believe that there are multiple. And part of it is also this divide between indica and japonica is very extreme. I mean, in many cases, they're not even cross-compatible. They're considered subspecies, but there's huge sterility barriers between them, and they are very different genetically. But at the same time, there's a lot of cross-flow, gene flow between them as well. So it's a very, I think, exciting crop to work with because of these reasons.
But I thought there were multiple domestication events within indica or within japonica. Is it just each one was believed to be domesticated separately?
Well, that's the controversial part because they've seen, like, even in Japonica, some of the specific polymorphisms they know came from indica and vice versa. Like in indica, like for the red rice, they know it came from a Japonica originally, this mutation source. And so it depends who you talk to as far as it's not really settled out yet, I'd say. But, yeah, right here.
Aerobic rice, it has shown that it will improve the yield. Is there any effort to conduct hybrid rice trials under aerobic condition? See what proportion yield improvement?
Right.
You can add extra, right?
Yeah, so
Hybrid rice increases yield by 15 to 20%, and aerobic another 20%, I guess.
yeah, it depends where you are. So I mean, in many areas, if you switch from irrigated to aerobic conditions, you'll actually lose yields. You'll have lower yields. But there's some breeding programs that are specifically breeding for aerobic, high-yielding aerobic rice, which, I mean, for people that aren't familiar, it's like growing rice like wheat in a way, is essentially not having an understanding in irrigation. In the past, we've had upland rice, but that's different. It's generally in the kind of mountainous areas, whereas aerobic is supposed to be high-yielding dry land cultivation. But I haven't heard, I think Arvind Kumar may be doing a little work on that, but I haven't heard too many people doing hybrid aerobic at the moment, just because hybrid has tended to just be irrigated so far. I imagine there would be good benefit from having a hybrid aerobic system. So, yeah.
Using the magic system that you described there, have there been investigations using heterotic groups in rice to diversify and understand the potential yield advantages of certain heterotic groups?
Yeah, that's interesting that you mentioned, I mean, combine the magic and the heterotic
Yes.
description, because in the past they've only looked at just, heterotic groups using markers, trying to define, essentially the genetic diversity relationship, and then intercrossing, what's the heterosis between these different subgroups. But I haven't heard anyone actually use the magic scheme as a way to test that. I think if you did like the magic and then maybe had test crosses to certain varieties, you could. But at Erie, they haven't been doing that. The magic was entirely for the inbred program. But I think it would be promising area of research. But I mean, the heterotic, so Fangming Xie was the head of the hybrid rice program, and he has done quite a bit on trying to define the heterotic groups. But and just recently they've had some good data, but it's still in the early stages, I would say. So I think we're about out of time. One last question, or is?
I have a non-science question.
Okay.
An opinion question.
Right.
Given and I guess this would be, was there gossip at Erie about this,
Right.
as to why Hank wasn't a co-recipient of the Peace Prize with Norman? Both doing the same thing, both major
Right.
crops, both dwarf, both millions of people involved. Any thoughts? Did the IRA come along and just 2 or 3 years too late for that to happen, or?
Yeah, I don't know the whole story, but I've heard that there is this feeling that Hank Beecher was kind of recognized, underappreciated, and that's, I mean, at Erie, of course, we know the history of how much he really did, yeah, I probably don't have much on the inside scoop on that, but I would certainly think he would certainly deserve it, based on IRA just had a massive impact in Asia, and so I think he should have, but I don't know why not.
A good chunk of that's probably political, right? Did
Oh,
Simmet have better
no,
lobbyists?
Right.
No politics.
That's the skeptical view. So, okay, well, thanks everyone.
Thank you.
Yeah.